Hey there, curious minds. Have you ever wondered how scientists assess biodiversity in different ecosystems? In just 3 minutes, we're going to dive into the fascinating world of environmental DNA metabarcoding. A cutting-edge technique that is revolutionizing the way we monitor and understand the diversity of life around us. Let's start with the basics. What is environmental DNA, or eDNA for short? Imagine every living organism shedding bits of DNA into the environment through their skin, mucus, feces, or other biological materials. This environmental DNA can be collected from soil, water, or air samples. Now, let's talk about eDNA metabarcoding. This technique involves amplifying and sequencing specific gene regions from environmental DNA samples to identify the different species present in an ecosystem. By comparing the DNA sequences to reference databases, scientists can accurately identify the organisms without physically observing them. So, what does the eDNA workflow look like? First, samples such as water, soil, or air are collected from the environment. Next, DNA is extracted from these samples, followed by the amplification of specific gene regions using polymerase chain reaction, PCR. The DNA sequences are then analyzed using high-throughput sequencing technologies, and bioinformatics tools are used to interpret the data and identify the species present. The applications of eDNA are vast and varied. From monitoring endangered species and tracking invasive species, eDNA metabarcoding is revolutionizing the field of ecology and conservation biology. Imagine being able to survey an entire ecosystem for its biodiversity in just a fraction of the time it would take using traditional methods. With eDNA metabarcoding, we can gain a comprehensive understanding of the species present in an environment and make informed conservation decisions to protect our natural world for future generations. And there you have it, folks. In just 3 minutes, we've explored the incredible world of biodiversity assessment and monitoring using eDNA metabarcoding. The next time you're out in nature, think about the hidden DNA signatures all around you, waiting to be discovered. See you next time.